A very warm welcome to you all, and I hope you're having a very good time at home. So, today's lecture topic is rabies virus, and we'll be starting with the learning objectives of this lecture. We'll be discussing the important properties, the transmission, and the life cycle of rabies virus. We'll be discussing the pathogenesis of rabies virus, and we'll be listing the clinical findings, laboratory diagnosis, preventive measures of the disease caused by rabies virus. This virus basically causes a disease which is known as encephalitis, and if we see in the book, rabies virus is a collective name given to many small viruses that comes under the umbrella of rabies virus. So to remember, what you have to remember is that rabies virus causes encephalitis, right? Okay. Now moving on further, we will be discussing important properties of rabies virus. It belongs to Rhabdovirus family. It has a single-stranded RNA enclosed with a bullet-shaped capsid. If you all remember what a capsid is, capsid is made up of many similar viral proteins known as nucleocapsid or capsomeres, right? So the shape of the capsid determines the symmetry of the virus. So the shape of this virus is the bullet-shaped virus. It is surrounded by a lipoprotein envelope. The genome of this virus is the RNA genome, or it contains RNA as the replicative genome in its structure. It has a negative polarity, and I am assuming that you guys remember what negative polarity was. Rabies virus has a single antigenic type, which is very important as far as the vaccination is concerned, because the more the antigenic type, the more the serotypes of the virus, the more different types of vaccine the scientists have to produce or make. The antigenicity resides in the envelope glycoprotein spikes, which are present on the outermost surface of the viruses. You'll see in the next slide in the picture. The important thing about this virus is that it can infect all mammals. जो ये पहले का एक myth है कि it can only infect humans. No, it can infect all the mammals regardless of they are humans or they are animals, right? With this, now in this slide you can actually see the rabies virus symmetry. As I told you that this is a bullet shaped virus, the capsid is of the bullet shape. You can also appreciate the glycoproteins, the spikes of glycoproteins. If you can see, these are the glycoprotein spikes, and this is where the antigenicity of the virus lies. You can also appreciate the envelope or the membrane of the virus. This light blue color, the pointer where I'm pointing, is the envelope, and you can also appreciate that after the envelope lies the glycoprotein. So the outermost thing is the glycoprotein spikes. Okay, you can also appreciate the ribonucleoprotein, which is the RNA genome, right? And the special thing about this virus is the shape of the virus, right? It is of a shape of a bullet. The replicative cycle of rabies virus is equally important as the pathogenesis is. So the rabies virus attaches to the acetylcholine receptor on the cell surface. Now if you recall where the acetylcholine receptors are, the acetylcholine receptors are present on the neuromuscular junction. So this means that when the bite of the animal will take place, the virus will straight away go to the neuromuscular junction and will target the acetylcholine receptor present on the neuromuscular junction. After it has gone to the new acetylcholine receptor, it gains entry into the cell, and this is where the virion RNA polymerase synthesizes 5 messenger RNAs that codes for viral protein. After the viral proteins are made, the next step is the replication of genomic viral RNA. After the replication of RNA, then comes the assembling of the viral protein. Now, the viral proteins which are made initially will assemble each other in a form of a nucleocapsid. If you remember, if you recall the basic virology lecture, you remember that there are capsomeres, there are many capsomeres which join together to form a capsid. So in the similar way, all the viral proteins that have initially been synthesized will assemble to make nucleotides. nucleocapsid or will assemble to make a capsid followed by the envelope. Now, how is the envelope acquired? The envelope is acquired as the virus buds through the cell membrane. Like for example, when the virus is coming out of the cell membrane of the cell that it infected, it will take away the cell membrane lipids and it will form its envelope from the cell membrane lipids. If we talk about the transmission of rabies virus, it is very important to note that the virus is transmitted by the bite of the rabid animal. Now, what are rabid animals? Rabid animals can be skunks, can be raccoons, can be bats, can be dogs, and can be cats. In our part of the world, that is the developing countries, dogs are the most common rabid animal that causes the disease to be transmitted in humans. If we talk about US, skunk, raccoon, and bats are the most common animals that causes the transmission of this virus. Now in US, if we talk about that the dogs are the rabid animals, there we cannot see that the dogs are the rabid animals that are involved in the transmission of this virus, because there the dogs are usually immunized or vaccinated. In the developing countries like India, Pakistan, and Sri Lanka, the prevalence of rabies virus is much greater than US because of one simple reason, that the dogs are not vaccinated here and not immunized here. Hence, the transmission of the rabies virus is very common in our part of the developing world because of the unimmunized dogs and cats. The virus basically multiplies locally at the bite site where the animal has bitten the human or any mammal. It infects the sensory neurons and it moves by axonal transport to the central nervous system. Now, if you think about this transport, or it is also known as the retrograde transport to the central nervous system, then the virus multiplies in the central nervous system and there it causes the disease known as encephalitis. When it causes the disease in the brain, which is known as encephalitis, it then travels down to the peripheral nervous system, to the salivary gland or to eyes or to any part of the body via these peripheral nerves. And then the animal is the human is basically showing the signs and symptoms of encephalitis. Just as we discussed that the virus, after traveling through the peripheral nervous system, can enter any organ or any gland of the body, so here it enters the saliva from the salivary gland and it can be transmitted again to any of the animal, to any of the human by the bite. Within the central nervous system, encephalitis develops with the death of neurons and demyelination. The infected neurons of the central nervous system contains an eosinophilic cytoplasmic inclusion called Nigri bodies. These Nigri bodies are basically the diagnostic marker for the rabies virus infection. So for example, a person who's infected with rabies virus कैन उसकी किसी भी किस्म की अगर हम बायोप्सी लेते हैं तो उसमें निगरी बॉडीज नहीं शो होंगी. निगरी बॉडीज आर स्पेशली सीन इन द ब्रेन बायोप्सी एंड यूजुअली this biopsy is taken in order to rule out the cause of the death of the patient, और ये पोस्टमार्टम रिपोर्ट में जो है वो हमें मिलती है. और अगर मैं लेमन टर्म में आप लोगों को समझाऊं कि व्हाट आर निगरी बॉडीज, तो ईजनोफिलिक साइटोप्लाज्मिक इंक्लूजन जो है वो हमें लाइट पिंक कलर की स्पेरिसिस में दिखती हैं. लाइट पिंक कलर की बॉडीज दिखेंगी हमें ब्रेन बायोप्सी के ऊपर. सो दीज आर द डायग्नोस्टिक मार्कर एंड अगेन दिस इज एन इम्पॉर्टेंट थिंग फॉर योर एग्जाम टू. So this slide will explain you the pathogenesis of rabies virus with the help of the diagram. If you see here, the first step is the viral inoculation from a rabid dog bite or a rabid animal. In this case, you can see that the rabid animal is the dog, and the dog has bitten the person, the human, on the foot. So from there, the virus replicates in the muscle of the leg, and the virus will travel retrograde through the axonal transport to the central nervous system. Now, if you see the step 3, virus binds to nicotinic acetylcholine receptors at the neuromuscular junction. I just told you that the acetylcholine receptors are present on the neuromuscular junction. The virus binds to them, and then it travels through the axons in the peripheral nerves, through the axonal transport, through the retrograde axonal transport to the up to the central nervous system. Replication, if you see the step 5, replication takes places in the motor neurons of the spinal cord, and ganglia travels to the brain. Now here in brain, infection of brain neurons leading to fatal inflammation. What is encephalitis? Encephalitis or encephalitis is the inflammation of your brain tissues. And by this we mean that the brain tissues have been inflamed and the person is now showing signs and symptoms of the rabies virus infection. We in the next further slides we'll be discussing what clinical signs and symptoms the person shows, but here we are just focusing on the pathogenesis of rabies virus infection. Now if you see here that the virus, after inflaming or after getting the brain inflamed, now travels again through the peripheral nervous system down to any organ of the body. It can either go to the orbit of the eye, it can also go to the salivary glands of the rabid animal or the humans, ठीक है जी, and there the virus enters and can be is ready to infect again any of the organism. अच्छा, अब ये जो एक मिसकंसेप्शन है कि सिर्फ ये ह्यूमंस में ही बाइट करता है, मैंने अपनी पुरानी स्लाइड्स में आप लोगों को बताया था कि द रैबिड एनिमल कैन इन्फेक्ट एनी mammal. So if for example अगर dog एक dog को दूसरे dog ने bite किया है and if that dog is having a rabies virus within its salivary gland, it can transmit the rabies virus to the another dog, right? And that dog can infect any human, can infect any other mammal also. Now we'll be talking about the clinical findings of the rabies virus infection. The incubation period varies from 2 weeks to 16 weeks, and I was just reading an article last week. It even showed that the incubation period of this virus can also last up to 1 year. So the initial phase, the prodromal phase, is basically comprised of fever and anorexia. It can even show as flu-like symptoms. So the people who have been bitten by any rabid animal does not even take into consideration that this can be the beginning of a deadly infection. So after the prodromal period comes the encephalitis phase, which comprises of seizures, delirium, and many other fatal signs and symptoms that the person shows after being bitten by the rabid animal. Now this rabies infection manifests as two forms, the furious forms or the dumb form. If you talk about the furious form में हमारे पास आता है encephalitis and dumb form में हमारे पास आता है paralysis या paresthesias आते हैं. अच्छा जी, एक और टर्म हमने देखी है, बुक में भी देखी है और आप लोगों ने अक्सर वीडियोस वगैरह में भी देखी होगी. Another term which is associated with rabies virus infection is the hydrophobia. As the name signifies, this is the phobia or this is the fear of having water. तो ये क्यों होता है, इसका रीज़न क्या है? इसका रीज़न ये है कि बिकॉज ऑफ द पैरालिसिस ऑफ फेरेंजियल मसल्स या फेरेंजियल मसल्स का स्पैज़म की वजह से द पर्सन हु हैज़ दिस इन्फेक्शन fears that when he or she will drink water, it will be very painful to engulf, or it will be very painful to drink that water past that pharyngeal muscle spasm. So that fear basically that fear or that pain basically provokes a fear in the person with this type of infection, and this is known as hydrophobia. So again, just a quick review of what the clinical findings may look like. Initial phase में there might be flu-like symptoms, followed by a proper encephalitis phase, which can either exhibit as seizures, it can either exhibit as anorexia or severe fever-like symptoms. And again, the two types or the two forms of this disease, rabies virus infection, are the furious forms and the dumb form. You will have to remember these things, which are very important for your exam point of view. For diagnosis, basically animal testing can be done, and this can be done because to check that the animal that has bitten the human or any other mammal is being infected with the rabies infection only, or is being immunized previously or not. Lumbar puncture, skin biopsy, blood test, head MRIs or CT scan are all part of diagnosis. But again, in our part of the world or in developing countries, जब तक diagnosis तक बात पहुंचती है, the patient has usually expired because they do not come at the point where he or she, where the patient has been bitten by the dog or any rabid animal. So as soon as the rabid animal bites or as soon as the dog bites, the protocol should be the person should report to the nearest hospital in order to check for his immunization and vaccinations. Now comes the most important part of the presentation, and this is the treatment and prevention of the rabies virus infection. So currently there is no antiviral therapy which is available for the rabies virus infection. So as soon as the person has been bitten by the rabid animal, he or she again should report to the nearest hospital and should be given a vaccine. Now the vaccine which is given to the person contains inactivated virus, and it is known as human diploid cells vaccine, short form as HDCV. Now human diploid cell vaccine is preferred, and this is preferred pre-exposure vaccine also, and this is also preferred post-exposure vaccine also. Now if we see the pre-exposure vaccine, now these pre-exposure vaccines are usually given to healthcare workers, are usually given to vets, are usually given to persons who are taking care of the animals in the animal rehabilitation center. That consists of 3 doses or 4 doses, and the doses are given at 0 days. For example, when the immunization starts, then after 7th, and then at the 7th day, then at the 21st or the 28th day. So this is the protocol for pre-exposure immunization. If you talk about the post-exposure immunization, which is very important also, this is when the person or when the animal is been bitten by the another rabid animal or rabid animal. This consists of basically the combination of two vaccines. Now one is the human diploid cell vaccine, which is known as HDCV, and the second one is the RIG, rabies immunoglobulin. And rabies immunoglobulin and HDCV vaccine should not be given at the similar spot in order, because this rabies immunoglobulin is the antibody. So this will obviously neutralize the inactivated virus given as a vaccine in the person for the immunization. So कहते ये हैं कि HDCV किसी और साइट पे दें और rabies immunoglobulin किसी और साइट पे दें in order to make the both vaccine highly competent and potential for the prevention of the disease. Tetanus vaccine should also be given just in order to avoid any form of tetanus botulinum or infection. So if you talk about what will happen when the person is bitten by the rabies infected rabbit animal, तो सबसे पहले wound cleaning होगी उस patient की, ठीक है जी. जब wound cleaning हो जाएगी तो फिर उसके बाद हम उसको post exposure immunization देंगे, which consists of 4 days और वो different days पे होती हैं. नेक्स्ट कम्स दी रेबीज इम्यूनोग्लोबुलिन जो कि हम उसको देंगे, ठीक है जी. अब एक और बात ये कहते हैं कि जिधर फॉर एग्जांपल रेबिड एनिमल ने बाइट किया हो ह्यूमन को, उसी के करीब जाके रेबीज इम्यूनोग्लोबुलिन दें. व्हाई? बिकॉज द रैबिड एनिमल विल इन्फेक्ट द वायरसेस, विल ट्रांसमिट द वायरस टू द बाइट साइज, एंड इफ़ यू गिव द इम्यूनोग्लोबुलिन देयर तो इम्यूनोग्लोबुलिन उस वायरस को न्यूट्रलाइज कर सकता है. So this is the theory behind giving the rabies immunoglobulin vaccine near the bite size, but it is not recommended to give rabies immunoglobulin vaccine near the HDCV vaccine, which is the human diploid cell vaccine, in order so that it does not neutralizes the that vaccine. So now we'll be concluding our today's lecture. सबसे पहले हमने बात की कि rabid animals कौन से होते हैं. Rabid animals हमारे पास होते हैं skunks होते हैं, raccoons होते हैं, bats होते हैं, dogs and cats होते हैं. In our part of the world, that is the developing countries, dogs are the most common rabid animals that causes rabies virus infection. So if we see here in this flowchart diagram, we can see that human and animal contact हुआ, मतलब rabid animal ने human को bite कर लिया. Now we have to figure out if the animal, is it a domestic animal or a wild animal, because उसकी वैक्सीनाइजेशन स्टेटस, उसकी इम्यूनाइजेशन स्टेटस से फिर हम जो ह्यूमन को बाइट किया है उसके ट्रीटमेंट और प्रिवेंशन की हम बात कर सकते हैं. Exposure हो गया, then comes the infection part. First it's the incubation period, which lies between 2 weeks to 16 weeks, and in some articles it is even written that 1 year, it can even lie to up to 1 year. Then comes the prodromal period. Prodromal period may you have flu-like symptoms, you have fever, you have anorexia, and the symptoms which are very vast and very basic symptoms, followed by acute neurological period, in which the disease can either present as the furious form, which is the encephalitis form, or it can even present as the dumb form or the paralytic form. So either ways it can lead to coma or death, and this is the end of the rabies virus infection. But if we talk about the treatment and prevention, we can see that prophylaxis can be given as a pre-exposure immunization and can also be given as a post-exposure immunization. In both cases, there are two vaccines which can be given: human diploid cell vaccine, which is the HDCV vaccine, and the another one is the rabies immunoglobulin. Now, rabies immunoglobulin should be given near the bite site, but should not be given near the HDCV vaccine in order to neutralize that vaccine. So it should be given near the bite site but away from the HDCV vaccine site. So this concludes our entire lecture of today. I hope it was very simple for you. If you have any question, you can email me your queries and I'll be happy to answer them. Thank you so much. Signing off from today.